So, hi everyone, it's my great pleasure to welcome you to the BIH lecture series, the online lecture translation from the Translation Hub Digital Medicine. My name is Kerstin Ritter and I'm very happy that James Cole is here to talk about the brain age paradigm. Before I'm going to introduce James, I would like to clarify some organizational questions. So we kindly ask you to turn off your webcam and the microphone at any time. I think this is done already. We will later have a discussion. You're very welcome to send your questions via the chat function. I will later ask the questions. The lecture will be recorded for our website. So here, therefore, it's also good to turn off your webcam and the microphone, and you are welcome to join us on Twitter under the hashtag BellInnovation and Charité Berlin. So it's my great pleasure to introduce James Cole to you. He is an Associate Professor of Neuroimage Analysis at University College London, based jointly between the Centre for Medical Image Computing and the Dementia Research Centre. He also holds a UK Research and Innovation Fellowship focusing on multimodality neuroimaging models of the aging brain. And he is one of the key authors who established the concept of brain age. James, I'm very happy that you're here and I'm looking forward to your talk. Thank you.
Thanks, Kerstin. Thanks very much for the kind introduction, and thanks very much for the opportunity to speak to you all today. The only sad thing is that, this is the sort of thing I would enjoy doing in person, but hopefully such things will be possible in the not too distant future. So I thought I'd start with a little bit of history, going back in time to November 1978. The New Scientist is a well-known international popular science magazine. Some of you may well read or even be subscribers to. I don't know how many of you were alive in 1978. I wasn't, but in this issue in November, they showed us the first NMR, as it was known then, scans of the human brain that were taken at EMI Central Research Laboratories. And you can see there in the right-hand corner, there is a picture, and that is a cross-section of the head of one of the research fellows in the laboratory who had their brain scanned in what we now call an MRI scanner. And this was some years after they first started building MRI, and they tried it on bits of the human body before, but this is the first brain to be published that I'm aware of. And as you can see, the quality is leaves something to be desired. But things have changed since then, as I'm sure you're aware. And if we compare 1978 to 1994, this was a scan from a 3 Tesla MRI scanner from the lab of Peter Mansfield. This is one of the first 3 Tesla scanners, and human volunteer and 3 Tesla scan. You can see very much now you're able to see the recognizable features of the human brain. Fast forward to 2019, this is a scan carried out at 10.5 Tesla on a human volunteer. There are no clinical systems of this field strength available, but experimental prototypes do exist. And you can see not only does the overall scan show great amounts of detail, including the vasculature of the brain, but if you see the inset where it's zoomed in, it's extremely high quality image. And you can really understand, and we've come a long way between 2019 and 1978 in terms of the things we can do with MRI to get images of the brain. And MRI has been an immensely powerful tool for research and for radiological practice. And this is a nice paper. It's a few years old now, but it's the history of MRI as per the journal Radiology by Robert Edelman from 2014. And you can see that, once these pictures of the brain started appearing, then the number of publications that used MRI or NMR and the number of publications in radiology itself kind of went up exponentially, up to the point that this paper was published, where there were 100, overall in the whole field, there were thousands of these papers being published every year and hundreds of them in radiology. And in terms of how radiologists work, the advent of MRI is a massively powerful way for them to see the human body and particularly the human brain, to help them make clinical decisions with patients with various different diseases. Of course, radiological practice is founded in the expertise and the skills of the radiologists, and often they are making a highly skilled but subjective qualitative decision based on the scans. If we look more broadly, this is a slightly more up-to-date version from actually did this morning. If we look for papers that are published on MRI and brain, we are topping well over 20,000 papers every year. And a lot of these papers are using various forms of neuroimaging analysis. And just picking two of the big main neuroimaging journals, Human Brain Mapping and NeuroImage, they're publishing more than 1,000, nearly 2,000 papers every year, looking at neuroimage analysis and how this can be used to take brain scans, MRI brain scans, and understand how how the brain works and how it's changed in disease and things like that. So there's a huge field of neuroimaging analysis research, and it's getting bigger. And I can, as MRI scanners become more widely available and cheaper, I only see it increasing. However, there are only really, in my experience, only very limited clinical applications outside of the qualitative radiological perspective. There are some available. These are mainly in the commercial domain, so medical image analysis companies that will, as a service, generate biomarkers from brain scans and supply them back to clinicians. And these have been used in clinical trials and various things. But in the UK, certainly, there are no approved NHS quantitative neuroimaging applications available to help patients. And for me, I see this as a really big bottleneck between research and clinical practice. There are, as you've seen, thousands of research papers going on in this kind of exploration domain, trying to understand how the brain works and use neuroimage analysis to understand brain diseases, but very few actually translate into affecting clinical practice. So why is there a bottleneck? What causes it? There's a great paper that I would recommend to you, written by Wu and colleagues from 2017, which is called Building Better Biomarkers, and that outlines what is this process that we need to go through in order to get into clinical practice, starting from small samples, going through to larger samples, where you have replications and things like that, and then looking at even bigger samples that go across scanners and different sites and things like that, getting to the point where you can use them in large-scale diverse populations. And at that point in 2017, they couldn't find any models that had got to this fourth level of translation, this population level. And that's really what we should be aiming for if we want to help, the patients with brain diseases. And part of it is illustrated in this nice mountain that the little figure is climbing on the left, where they're moving from exploration, demonstration, characterization, all the way through to surrogacy, and it gets increasingly hard, and the investment required increases as you go through. So the bottleneck is caused by a lack of time. This process to go through to population level neuroimaging takes time. It's expensive. Another issue is that we may aspire to doing interdisciplinary research, but actually it's quite easy to have a whole career where you just focus on one area and you don't necessarily try and translate your results, from computer science, for example, into medical practice. There are also potentially, I would argue, a lack of incentives for people to do this. Although there are funding mechanisms, translational funding mechanisms around, it's still quite possible to be be very productive in scientific research, but without getting to the point of translation. And I think if there could be more incentives around that, that would help. But overall, the biggest bottleneck is it's a difficult thing to do. There are lots of steps, and actually finding the models and the neuroimage analysis pipelines that can work in clinical practice is hard. But I think for me, it's a very important challenge. So I'm going to talk to you today about what I've been trying to do towards clinical applications of neuroimaging analysis, and particularly I'm going to talk about my work using the brain age paradigm. And as Kerstin said, I'm fortunate enough to be based between two different places at University College London, both the Dementia Research Centre and the Centre for Medical Image Computing, and this actually puts me in a good opportunity to try and do some translational computer science, neuroscience into clinical practice. And I'm going to talk about the work I do at these centres today. So most of the focus of my research over the last 6 or 7 years has been on this brain age paradigm. Some of you may have heard of it, but for those of you who haven't, I will explain it, what I understand by the brain age paradigm now. So to understand brain age, really what we're asking is, how old is your brain? But actually, when you're asking that question, you really need to pose a bigger question, which is, what is aging? If you're asking how old is something, what is aging? This is normally the kind of thing where actually I would sort of try and get some audience participation going if we were face to face, to get people's ideas of what they think aging is. And over the years, I've had some very interesting responses. But in the absence of that in the online format, we'll start with a well-known repository of definitions, which is the Oxford English Dictionary, which very concisely defines aging as the process of growing old. While this is concise and hard to argue against, it doesn't really tell us very much about what that actually means. So a better definition comes from Sir Peter Medawar, the Nobel Prize-winning biologist, who said that aging is the collection of changes that render human beings progressively more likely to die. Now, this is a very morbid definition. I don't know if Peter Medawar was a morbid person particularly, but it certainly suggests that aging is around the progression towards death. Denham Harnham, who is one of the sort of doyens of biogerontology, he defined it as a bit more verbosely as the result of progressive accumulation of changes in the body which occur with the passing of time and which cause the increase in the probability of disease and death of the individual. So along with similar lines to Medawar, Harman talks about both disease and death, but also focusing really on the biological changes that happen with the passage of time. So the simplest way to measure aging is literally just the passage of time, and it's clear that there's lots of evidence that suggests that your risk of dying indeed is increased once you hit adulthood. Your risk of death is relatively linearly increased with the passage of time. So clearly aging and death are intimately linked. And we know one of the major consequences of aging is there's the increase in disease prevalence. And this can be heart disease, this can be brain diseases, this can be cancers, all of which will become increasingly prevalent as people get older. But this must be driven by some biological process. So what is biological process aging? Can we define it from a biological perspective? Well, we can certainly try, and lots of people have done this. There was a canonical paper that came out in 2013, which was the result of a meeting of many of the leading biogerontologists at the time that defined this kind of this paper that set out the various different hallmarks of the biological aging process. And really, it seems to be that the hallmarks themselves kind of reflect almost every biological system in the human body. And that can be from the sort of organ level through to organelles like mitochondria, but also at the protein level. So proteostasis breaks down, and cells can senesce, but they can also, the communication between cells has changed. And even at a molecular level, you see changes to telomere loss, epigenetic changes in histone modification and DNA methylation, and genomic instability with replication of genes. And basically, the aging affects everything, and either directly or indirectly. So although that could be seen as bad news for the human body, it's also good because it gives us lots of different approaches to actually measure the aging process. And what we do know is that aging in humans is universal. There's nobody who doesn't age, but actually it doesn't affect everyone in the same way. This is someone who was diagnosed with a very rare genetic disorder called Hutchinson-Gifford progeria. Progeria literally means more aging or accelerated aging. And you can see, so this person was 17 years old when this photo was taken, and you can see that outwardly he's exhibiting lots of the sort of phenotypes that you might associate with older age, such as skin wrinkling and hair loss and loss of teeth. And unfortunately for people with this condition, the life expectancy is much much reduced. So clearly genes, in some cases anyway, can have a big impact on how your body ages. But it's also true that environments can have a big impact on the aging process. So potentially people who are exposed to adverse environments could experience some of the age-associated changes more rapidly than you might expect. But it's not all bad news. This gentleman, his name is Fawaj Singh. He's famous in the UK for being the oldest man to complete the London Marathon. He only took up running when he was in his 80s after his wife died, but then he ended up running the marathon when he was 101 years old, and he is still alive. I think he's 112 now, but I don't know if he's still running marathons. I think he focuses just on 10 kilometer races instead. But so clearly aging doesn't affect everyone in the same way, but it's not always bad news. But this variability is important because it gives us a way of measuring biological aging. There are lots of different approaches that have been taken in the field to measure biological aging. Some as basic as, how many teeth do you have, or how healthy are your gums, with the idea that, the older you are, the worse your dental care is. But more sophisticated things include physiological measures like grip strength, molecular measures like telomere length, or protein expression of proteins associated with cellular senescence. You can even measure the proportion of your CD8 T cells that are positive for cytomegalovirus, with the idea that you might get infected with this relatively, usually benign virus more frequently the older you are. Perhaps the leading biological measure of aging is the epigenetic clock, which uses signatures of DNA methylation in the CPG sites across the genome to predict how old people are. And you can use blood samples, or you can use any other tissue and look at the DNA methylation patterns and get very accurate predictions of chronological age from DNA methylation chips. However, so those are some of the ways of measuring aging. But I'm particularly interested in the brain, and being a neuroscientist, I want to understand how aging affects the brain, and why this is, and what are some of the consequences of that process. So the main consequence really of brain aging is cognitive aging, and it's clear there's an abundance of research from psychology and cognitive neuroscience that shows that cognitive performance decreases as we age, and this affects almost all domains of cognitive performance: memory, attention, concentration, information processing speed, executive function. There are a few exceptions, such as vocabulary, world knowledge, but generally speaking, as time passes, there's a moderate but steady decline in cognitive function after we hit adulthood, up to our 80s and beyond. And age-associated cognitive decline is important because it's associated with a number of outcomes, such as decreased functional capacity and ability to work. It can lead to quite significant decreases in quality of life, and at a societal level, it's can lead to increased care costs as well, to look after people who are suffering from cognitive decline. And perhaps most importantly, people with pronounced cognitive decline are also at increased risk of developing dementia. And there's a sort of clinical entity known as mild cognitive impairment, which is often seen as a sort of key pre-manifest stage of dementia. Particularly, there's evidence that shows that people with mild cognitive impairment are at perhaps 8 to 10-fold higher risk of developing Alzheimer's disease. It doesn't mean everyone who has mild cognitive impairment will get dementia, and nor is it the only route to dementia, but it is a key risk factor. So if we can understand what causes cognitive decline, this could have a big impact on a number of different fields. And clearly, the cognitive decline is caused on some level by changes to the brain. So what are the changes that happen to the brain? At a macroscopic level, the brain gets smaller, it shrinks. People's early studies into brain aging used post-mortem samples and weighed the brain. The brain of older adults is lighter than younger adults. This is manifest in ventricular enlargement, thinning of the cortex, and atrophy of the subcortical regions. You also see these characteristic infarcts in the white matter, which cause cause lesions, and some people refer to them as white matter hyperintensities because of the contrast you get on T2 weighted MRI scans, which accumulate with age. You also get cellular changes, so neurons themselves will shrink and eventually die. Synapses get pruned, and axons will degenerate and release byproducts into the blood circulating system. You also see in the brain patterns of protein deposition. The two most commonly reported are beta amyloid and tau, these plaques and tangles that are so often associated with risk of Alzheimer's disease. Although it's important to note that actually people who are perfectly cognitively normal will also can have high levels of misfolded proteins in their brain. So clearly, you know, it seems to be part of aging, and it may well increase risk for dementia, but it's not 100% predictive of it. And maybe these things are actually more general rather than specific for forms of dementia. We can also measure how the brain changes with age using MRI, and that's one of the techniques that has been used very commonly. This is a few years old now, this paper, but it's a very nice overview of the volumetric trajectory based on whole brain volumes of people throughout their lifespan. And you can see on the left-hand side here, they've got all the lots of data points plotted that shows that brain volume, after you hit adulthood, your brain volume decreases in a relatively linear, moderate way. And on the right-hand side is the growth rate. So again, once you hit adulthood, you hit this kind of negative growth rate, which means your brain is essentially getting smaller. And there's lots of data to support this as a very solid finding. However, when you start looking into different brain regions, the patterns are a bit more variable and a bit more non-linear. So this is work from last year from Christos Stavrotikos' group that shows that if you take different regions of the brain, including the hippocampus, the thalamus, some of the deep grey matter, or the frontal grey matter, they actually show differing trajectories of change over the lifespan. And actually these trajectories may well be non-linear. And you'll have different areas of the brain which show patterns of exponential change followed by plateaus and things like that. So, and you can see also here we've got the ventricles on the top left, which is the one, so the ventricular spaces are enlarged. But again, this is perhaps not linear, and the timing that these changes take place may well not be the same in different parts of the brain. So although the whole brain is affected, it's not affected in a really predictable, uniform way, and it can be spatially variable and non-linear in nature. So now that we know that the brain changes with age, and we want to try and measure that in some way, we need to come up with a model. So I'm going to talk about my approach to modeling brain aging. And this is predicated on the idea that as we get older, even if you have a healthy brain, you are gradually getting closer to some sort of symptomatic threshold that is a risk of cognitive decline. However, there can be events that happen that put you on a different trajectory. This could be an environmental insult, it could be accumulative insults, it could be a genetic thing, and you can be on various different trajectories from a sort of healthy aging trajectory. And these trajectories are important because the steeper the trajectory, the earlier you are going to cross this threshold for showing symptoms of cognitive decline. So the hope is by characterizing healthy brain aging, we can then measure an individual's difference or distance from that healthy trajectory to get an idea of what sort of trajectory they are on as an individual and how likely they are to have symptoms of cognitive decline or other age-associated diseases in the near future. So this is one way of actually doing the modeling itself. Once we've got the model, we need some data to fit a model. And the approach that we've taken is to try and collect a very large, healthy training data set of brain MRI scans of people whose age we know. We then typically do some image processing and then use a machine learning or multivariate regression model to predict age as the outcome measure. When we've done that, we can then validate the model using either held out or in this case cross-validation, where you remove a subsample of your data and you try and predict the age of those individuals based on the brain features that you put into the model. you know, that you've trained on the remaining 90%, for example, if you're using a tenfold cross-validation. When you're happy that your model is giving you reasonable performance, you can take the coefficients of the model and then apply them to any new data. And that will then give you an age prediction. And then the final step is to take that age prediction and then compare that to the individual's real age. The idea being if their brain predicted age or brain age is greater than their chronological age, that suggests their brain appears older than you would expect given their age. And likewise, if their brain age is younger, then that suggests their brain is appearing younger than you would expect. So that's the kind of framework that we use to predict these things. And we've done this in lots of different ways, but I'm going to mention one particular example of some publicly available software that we've got that was trained on T1 weighted volumetric MRI data from 7 different public repositories. And we accumulated a training data set of over 3,000 adults aged 18 to 92 with an average age of around 37, approximately evenly split between males and females. These images were pre-processed using some standard voxel-based morphometry tools like tissue segmentation and nonlinear registration, and then resampling and modulating into a common space to provide these 3-dimensional maps of brain volume. And then we were able to put that into a Gaussian processes regression and get very accurate predictions of chronological age. And this plot you can see on the right-hand side is a real age on the x-axis and predicted age on the y-axis. And you can see that we do a very nice job of predicting approximately how old people are. And using held-out test validation, we got a mean absolute error of under 4 years. So we're able to explain well over 90% of the variability in chronological age from brain features alone. We tried this in a completely independent sample, and again, the performance was slightly worse, but it's still very strong. And this software is available for anyone to download and use. It's called BrainAger, or BrainAge R, because it's mainly using the R software package, and it's on GitHub there. So we then, now that we have this model of age and we can show that it works accurately, we want to understand how well does it actually recapitulate other things that happen in aging. And we were lucky enough to collaborate with people at the University of Edinburgh to get access to the Lothian birth cohort 1936, which is a study of adults born in 1936, as you might guess. There were over 600 of these people who all had an MRI scan at age 73. And here you can see what they're particularly interesting, I think, is probably the histogram on the right hand side, where in red are the real ages of those individuals. So you can see, because it's a narrow age cohort, that red plot is very red, those red bars are very tight. The blue bars are the predicted ages based on their brain data. And you can see the variability is much, much wider in brain predicted age than it is in their real age. And what we wanted to do is use other information that we had on these people and see whether that variability relates to other facets of the aging process. And indeed, we were able to see that yes, it does. So people with older appearing brains had poorer fluid cognitive functioning, weaker grip strength, lower lung function, slower walking speed, and an increased allostatic load. Allostatic load, for those of you who aren't familiar with it, is a composite measure of biological and physiological parameters that are supposed to reflect the sort of physiological wear and tear of environmental adaptation. These include things like body mass index, cholesterol levels, lipid levels, blood pressure, HbA1c levels, and various other things. So the data from this study really helped support the idea that actually our brain measure of aging is related to other ways of measuring the biological aging process. Particularly interesting in this cohort, we were able to see that actually, because this cohort was followed up over a 7-year period after the scans were taken, we were able to see that those who had older appearing brains at baseline actually were at increased risk of dying in the follow-up time. So you can see the Kaplan-Meier plots on the right-hand side show you that suggest that those with a higher brain predicted age difference, or brain PAD, are actually a considerably greater risk of mortality than those with the younger appearing brain at the time of their MRI. So this really suggests that this measure is picking up something quite general about the body's health, because these people didn't die of neurological complaints. It was mainly cardiac issues that was the cause of death in these people. Particularly interesting as well was that we were able to access epigenetic clock data on the same cohort, because they'd had that process done already, and we saw that there was actually no correlation between their DNA methylation age and their brain age. However, when we predicted mortality in various different models, we found that although both DNA methylation age and brain age were independently predictive of mortality risk, the strongest model that explained the most variance was a model that combined both of them, which suggests that although unrelated statistically, they are explaining some complementary variance in terms of biological aging. And this for me is suggestive of evidence that we should try and measure biological age in lots of different ways to come up with a more comprehensive way of understanding the aging process, rather than just using single measures. Of course, the more measures you have, the more complicated a study is. But now that we're reasonably happy that our brain age measure is actually picking up something that is age related, we then chose to look at brain age in the context of different diseases and really try and answer the question or provide evidence towards whether these different diseases are actually related to acceleration or added aging of the brain. And indeed, that's what we found in lots of different contexts, actually. So we've seen that people who have suffered a traumatic brain injury, people with HIV, people with medically refractory epilepsy, people with Down syndrome, and people with multiple sclerosis, and even people with major depressive disorder all have older appearing brains to a lesser or greater extent. The difference in major depressive disorder was less than a year, but the difference in multiple sclerosis was about 10 years. So the average magnitude of the brain aging effects that we've observed in these diseases is quite variable. But really, it's that variability at the individual level that is particularly interesting for predicting outcomes, and that's what we hope to use in a clinical context. And so that moves me on to talk about how, if we have this brain age measure that is sensitive to different diseases, how can we use that in the clinical practice? And I'm going to talk about 3 different studies where we've tried to, or 3 different approaches to doing this in real-world contexts, using hospital data and trying to influence radiological practice. So I'll start talking about the real-world context. And this is a study that we call the Barcode Study. And we were fortunate enough to have access to a data set that was acquired from a number of different memory clinics in South London that were sent for routine clinical MRI to a research department where they had high resolution 3 Tesla volumetric MRI scans done. And this is a study that accumulated over many years, and we ended up with around 4,000 people who'd been to memory clinics, who'd all experienced some kind of cognitive symptom that led them to be referred for an MRI scan, having been to the memory clinic. So this is very much a real-world data set. There were no exclusions based on comorbidities or ethnicity. South London is generally a highly ethnically diverse part of Britain, and also very economically diverse as well. And these people were referred clinically, so there's no recruitment bias and there's no sampling bias in this data set, which is a big issue with a lot of research studies. There is this research volunteer effect, which is hard to overcome. So this data set was really literally taken from scans that were used in clinical practice. And the other great thing about this data set is that they were linked with electronic health records. This is a system that's set up between the NHS in London and King's College London, which is where it was based when I carried out this work. And the study has been led by my colleague Francesca Biondo, who's done the analysis on this. So we were able to use this linkage to electronic health records to find out what had happened to people after they'd had their MRI scan. So we restricted the cohort to people who had not received any kind of dementia diagnosis within 3 months of their scan, so that we were making sure that our study was looking prospectively rather than contemporaneously. And that got us down to just over 1,100 people, and we ran the brain age paradigm that I've described to you earlier on the MRI scans of these individuals, and we were able to do a survival analysis looking at their time to dementia. And for those people who didn't go on to get dementia, they were censored based on their time of last clinical contact or last diagnosis of something that wasn't dementia. And we saw that there was a significant effect of their brain age on their survival before getting a diagnosis of dementia. And this was true when accounting for age, mini mental state, exam score, and normalized brain volume as well. So for every 5 years of increased brain aging, there was approximately 15% increased risk of having dementia diagnosis in the study period. We did a couple of sensitivity analyses as well to make sure that we were, to get a better idea of what we're tapping into. So we limited the study to the 250 people who had an MMSE score of over 27, so suggests that they're really cognitively normal. And actually the effect of brain age in this group who wouldn't present with any really strong clinical symptoms was even stronger. We then also limited it to the 500 or so people who did not receive any diagnoses within 3 years of the scan, and to try and see how long term these changes, how far in the future we can predict things. And the effect of brain age as a significant predictor here was the same as the main analysis. So that remained robust even when changes were happening several years after the MRI scan. So this really suggests that taking data that can be very messy, very dirty, include people who've been misdiagnosed and misreferred, and people from all sorts of different backgrounds that is not stratified based on the research criteria, we can use brain age in a clinical context, and it provides useful information over and above the sorts of information that people have already. This was a special study though, because we were able to get these 3D, three-dimensional volumetric MRI. Most hospital scans do not include that in the regular protocol unless there's a really strong indication of dementia. So the next study I'm going to talk about is a big study I've also done with King's College London using hospital data. So we were able to collect retrospective data set from two very large hospitals in South London, and we had access to these typical clinical scans that were T2 weighted MRI or diffusion weighted MRI. And in this picture you can see the difference between the sort of research grade volumetric scans at the top and the clinical grade T2 and diffusion weighted scans. The in-plane resolution is very similar, but as you can see looking at the sagittal and coronal slices, the resolution is much worse. So the slices are about 5 millimeters thick, and they are much coarser, so you have much less information to work with. But this is the sort of thing that's collected typically in a hospital, and this is the sort of thing that you'd want to work with if you're going to apply this in a clinical context. So we were able to use a very nice system. So this was developed by my collaborators Tom Booth and David Wood at King's College London. They have a very nice system where they use natural language processing to scan through radiological reports and identify people who had no pathology or were reported as normal. And I found 23,000 of these people and used a different, so this was using a different approach to the previous brain age model I mentioned, where we have a dense net convolutional neural network to predict age. And we were able to show that you can get very accurate predictions of chronological age in these people. So we had these two different hospitals, and we found that you can still get accurate predictions if you train on data from one hospital and predict on another. We also tried to see whether you could train on diffusion weighted imaging and then predict on T2 and vice versa. And that's on the right hand side here. You can see these red blobs are the cross modality predictions, which doesn't work, which is a shame, but actually the within modality predictions are very strong, and we're getting very low mean absolute error and explaining lots of the variability. The other great thing from this study was that we were able to identify more than 200 people who had radiological reports that they were that these patients had excessive atrophy for their age. We were able to then run those as a test set through our trained model, and you can see the results here in the plot. These are the red points where they had, on average, nearly 6 years older appearing brains than their real age. So this suggests that when the radiologist is making a qualitative decision of whether this person has atrophy that's excessive for their age or not, we're tapping into the same signal with our quantitative, objective, and automated pipeline that we've developed for this, which really shows that the brain age models are picking up something that does have clinical relevance. So you know, this study really shows that with hospital data, you know, we can still do something that could actually have an impact at a clinical level. The final thing that I want to mention is about how we've been trying to work with radiologists to influence radiological practice. And this is the results of the Quantitative Neuroradiology initiative, which is run at UCL, led by people such as Frederick Barkov, Tarek Yousri, and I've been working with Olivia Goodkin and Hugh Pemberton to try and implement this. And they've done all sorts of work behind the scenes in generating automated brain image analysis pipelines that will generate summary measures that can then be put in the form of a PDF with images to then be sent to a radiologist to help inform them further about that person's clinical state. And these are a few examples. We have specific pipelines for dementia, specific pipelines for multiple sclerosis, specific pipelines for hippocampal epilepsy, and the idea is that we will include in these reports the brain age of these individuals, so that when a radiologist is tasked with making a decision about you know, the clinical decision for a patient, they can actually refer to their brain age and see how that relates to their real age, and then use that information to hopefully help improve their decision making. And the plan is that we're going to run a qualitative study where we actually interview, we provide a sort of scenario where we have a group of radiologists who are given example cases with and without various bits of information, and ask them to kind of rate whether they find these bits of information useful. And the hope is that they will find them useful, and that we can go from just, you know, not just being statistically informative, but also practically informative. And if that's the case, then the hope is that, you know, there will be calls for these things to be adopted in regular clinical practice in the hospital here. And if it can get adopted here, then, you know, we can potentially roll it out to other hospitals around the UK. So in my efforts to get towards a kind of clinical adoption of brain age, using neuroimaging, hopefully I've shown you that how old your brain is or how old it appears does matter, and that might not be the same as your chronological age. Neuroimaging can be used to measure the brain aging process, and that neurological and psychiatric diseases can actually influence this process. In my view, the brain age paradigm could be a useful general biomarker of the aging process, and that it, you know, has the potential to provide information about age-related health outcomes. And I hope that, you know, the work that I'm doing is showing the potential for this to be deployed in clinical context. There's still a lot more to do, but hopefully the kind of studies we've been running recently show that it is possible, and that, you know, with more work we can actually get through the bottleneck. And just to finish, with a more recent copy of the New Scientist, this is actually from 2019, and I was very flattered to actually be asked to be interviewed, and this was when I was at King's College London, but yes, I was very, very flattered and grateful to have the opportunity to have my name appear in the New Scientist, even if it's 30 years after that amazing brain scan from 1978. So that's it. Thank you very much. I'm very happy to take people's questions.
Thank you very much for this awesome lecture. Really interesting work, very nice data sets. Thank you very much. So you're all invited to post some questions and I'm going to read them. So maybe...
Should I stop sharing my screen?
Yes, maybe stop sharing. So I have a first question. I think it was very interesting that you showed like many different diseases where brain age plays an important role, and I would like to ask if there are like specific alterations in the brain which are more specific for MS or specific for depression. So because you have just one kind of summary marker, and can you also explain that, for example, MS and large ventricles are more important, and for some others maybe hippocampal atrophy or so? Do you have some results or ideas on this?
Yeah, so the brain age paradigm is designed to be quite a general marker. So it's looking at information across the whole brain and integrating that into a single number. And that means potentially you could have increased atrophy in one area or another and still end up with the same brain age. So it could well be possible that the disease specific patterns that you see are overlapping with the general aging pattern in different ways, but that still leads to an increase in brain age. So there are lots of different ways we can approach this. One thing that we've been doing, I didn't talk about it today, is we've actually been trying to develop a local brain age model where you actually get a voxel-wise, so the fine grain resolution prediction of brain age. So then you can develop generate an individual map of someone's brain age across the brain to look at variability. You can obviously then take those maps within a disease group and average them to see whether there are consistent patterns of increased aging in the hippocampus or the ventricles or the frontal cortex, depending on different disorders. So we've actually got a paper that's under review at the moment where we've done this in dementia, and it does seem to work quite well. And the hope is that we can roll this out into other conditions later. But yes, that's the goal, because certainly, to get mechanistic understanding of what's going on, this more fine-grained spatial resolution will be really important.
Yes, thanks. Interesting. And with respect to the very different data sets you analyzed, and it was also very nice that you also really analyzed clinical hospital data. So how much effort was it to pre-process the data or to harmonize the data? And
so for the hospital data, it was a lot of effort for somebody else. I was very fortunate to work with some great collaborators at King's College London. Tom Booth is a radiologist, and he led the application to get access to this enormous retrospective data set. So that in itself, just getting the ethical approval and the logistical, access to these data was a huge effort. And then David is a research fellow who's in Tom's group, and he did a lot of the pre-processing and data management of that, which again has been very effortful for him. I've been fortunate to just sort of advise on the brain age side of things and help with statistical modeling. So yes, I think if anyone else is trying to do something similar with a big hospital data set, it can be very hard work, but actually it's extremely rewarding because it provides a very big resource that you can use for lots and lots of different things. And particularly the great thing about David and Tom's setup is they've used this natural language processing so that you can sort of parse the radiological reports. So if you want to find a subgroup of people from within a big hospital data set, as long as you've got the image and the radiological report, you can use this framework to find people who have a stroke or have a brain tumor or have who are healthy or have no abnormalities and things like that. So we've really used that, and that's made it much more tractable. Otherwise, you would need to have these things labeled manually, and that's extremely onerous process as well. Or you would need to go through and read all the reports yourself, which is just not feasible. So the pre-processing, in terms of the image pre-processing for the hospital data, is relatively limited actually. Things like intensity normalization. We deliberately try to avoid doing intensive image pre-processing because we hoped our convolutional neural network model would be able to learn the key features anyway. And we wanted to avoid the time and the kind of potential errors that can come from image pre-processing.
So you did not do any spatial normalization into MNI space, you just left the images out. And this is also the main reasons why you use CNNs here for the hospital data. Yes. Do you have some, would you generally say CNNs are better for this kind of task, or it really depends on the data set?
I think if it's tractable to sort of hand engineer the features, then I don't think CNNs give you quite such a boost. But in these contexts, then where you're trying to learn something a bit more implicitly, then I think they are very powerful. I would say you do need bigger data sets, and that's why we were very fortunate to be able to get 23,000 healthy people. It'd be very hard to do that prospectively, but because we were doing it retrospectively, it gave us lots of power. So this is a case where CNNs allowed us to do something that we couldn't have done with a more conventional machine learning approach.
Thanks. So there are two questions from the chat. So the first one is, how do you interpret the finding that DNA methylation and brain PET appear to be statistically independent, nature versus nurture?
That's a great question. I would say to take that result with a bit of a pinch of salt, because it's a narrow age cohort. It's possible that if it was a much bigger age range of people, we may have seen more shared variability. So it just happened to be this snapshot at age 73 and we saw there was no relationship. So it's hard to say that there's never a relationship outside of that range. Yeah, I think it could be two unrelated sets of environmental processes. Obviously, with the DNA methylation, as I said, you can get this from any tissue. It's often done from blood because that's one of the most accessible tissues. They have done post-mortem brain tissue DNA methylation aging, and that can be done accurately. But obviously, getting data where you have in vivo MRI and post-mortem tissue of the brain is very, very difficult. So it's possible that were we able to get the DNA methylation age from the brain, it might be much more similar to the brain age. So it's quite hard to draw firm conclusions on that.
Thanks. Next question, did you analyze whether the treatment for the specific diseases you looked at had an effect on brain aging as well?
That's a really good question. So these were all retrospective studies, so we haven't ever had the opportunity to do a kind of prospective treatment type trial with brain age as an outcome measure, which I would love to do. But for example, in the major depressive disorder study, where we showed a significant but moderate effect of brain age, the presence or absence of antidepressants did not have an effect on brain age. In the multiple sclerosis study, we were able to see whether we had a sort of binary, does the individual receive disease-modifying therapies or not? And again, that didn't have an effect. But unfortunately, those are very, very coarse ways of measuring what is actually a very complicated phenotype, because obviously all these treatments differ, and how long the individuals have been on the treatments will differ as well, and dosage can have an effect. So, we haven't found evidence that treatments do affect brain age, but that doesn't mean that they don't. We just haven't been able to look properly, really.
Thanks. Two more questions here, which are related. So first, what is your suggestion on the neural network to use? Complexity is varying, and there is an ongoing discussion about that a very complex network, it's not necessarily needed to predict complex relationships. In that regard, features you extracted to train the network is important. Would you comment on what you used? I think this refers to the CNN from the...
So I completely agree that, network complexity is not necessarily a good thing. And in fact, really, we should be quite judicious as we add more parameters to our models, because obviously the more parameters you have, the more likely you are to overfit. So really, you want to start with the most simple model and then build up and only add complexity when you can show that it's actually beneficial to do that. And that also has an impact on how interpretable these are. The dense net that we used in the context of the specific hospital data set that we were using was actually taken from a very nice package of open source neural networks designed for medical imaging called MONI. and that was a dense net that is quite complicated and has got a lot of layers. But that was more for the expedience rather than trying to spend too long optimizing the architecture. The idea was, let's take something off the shelf and see how well it works, because I think when you start doing a lot of network optimization, then you open yourself up to the criticism that you've just sort of fiddled until you got a good result. Whereas if you can take something that is publicly available and anyone can use and just change that as little as possible, and then still get good results, then that's a bit more transparent, even if it's not following that sort of that heuristic of trying to be as uncomplicated as possible. So yeah, it's a bit of a balancing act, I would say. In terms of the features, it generally seems to be quite widespread, but maybe more ventricular and subcortical than cortical, I would say.
Okay, thanks. Then there was a question on the impact of the training population's age distribution on the brain age model. So one model has been trained on the age group from 18 to 92 years with a mean age of 36 years. Does this bias the estimation for older individuals? Does it work the same way for all individuals?
That's a really good question. So the types of regression model that we use, so for example, Gaussian processes, is very sensitive to local distributions of data. So actually having an uneven distribution in the training data set wouldn't cause a bias. Actually, and we've looked at this quite a lot in various different ways to check that wasn't the case, and indeed it's not. I think if you were just using a linear regression, that could be a big problem. But with these more non-linear probabilistic models, then they're much more sensitive to different distributions, local differences in distributions. The availability of healthy training data of older individuals is harder to come by, so we have fewer examples. So I would say that the predictions are slightly less accurate in older people because we have fewer training examples, but that's not because we have lots of examples at an early age, it's just because we have fewer examples at an older age. So it's more of a noise than a bias thing, I would say.
Okay, so there's some more questions. Maybe could you comment on how stable you think brain age would be across different sites and scanners?
Yeah, so that's obviously a really important issue. So the most of the brain age work that I presented, not the hospital stuff, but everything else, has all been using training data sets from lots of different sites. And that, by having what I sometimes refer to as dirty training data, actually makes them more robust to new data sets. So you're learning something that is patterns that are common across your training data set rather than overfitting to specific data sets. And the fact that when we used a completely independent test data set, we saw a slight decrease in performance, but not a big decrease in performance, suggests that it does generalize very nicely. We've also got some small data sets where we've had the same people scanned on 2 different scanners, and the results are very reproducible between them. So the interclass correlation coefficient suggests the between scanner reliability of the model is very high. We haven't done that very well in the hospital CNN model. We've compared it between the 2 hospitals and it works very nicely, but we haven't gone further and looked at other data sets with that yet. But that's the plan. Okay,
so 2 last questions very shortly. So one is more about communication. So for the 1936 cohort study, you told us that there are huge differences between estimated brain age and the actual age, and wouldn't it be maybe more pleasant to patients to report the differences in categories and not in years?
So that's a really interesting question. I think one of the reasons I've been attracted to this brain age paradigm is that it has a kind of intuitive public health message, and actually in the UK anyway, doctors use things where they talk to patients, they ask patients about their cardiovascular health, and then in a very, very sort of qualitative way, they kind of calculate their heart age, and based on do you smoke, how much exercise do you do, that kind of thing, and they would say, well, your heart age is 10 years older than your real age, and that makes people change their behavior, actually. So my hope is that if somebody was told your brain age is 10 years older than your real age, you should lose weight, stop smoking, do more exercise, then that might actually, rather than saying your hippocampal volume is in the 32nd centile, then actually having something that they can relate to about this kind of age, in real terms, then that would actually be a strong public health message, actually.
Okay, so they're coming again, some more questions.
I would say if anybody's got any questions, very welcome to email me and ask me after the session. I'm very open to that if we are running out of time.
Yes, yeah, this is a great offer. Thanks, James. So it was really interesting, and it was really nice that so many questions and so many interesting questions
thanks
came.
again for the invitation, Kerstin, and thanks for everybody's interest in the talk. As I said, please get in touch if you've got any follow-up questions. Very happy to discuss further.
Yes. Okay, thank you very much to everyone, and thanks again to James, and have a good day. Bye.
Bye-bye.